welcome again, Professor Umapathy, in this first 2022 session of ACS Science Talk.
Right.
So we'll begin with a couple of instructions and again, the updates around ACS Science Talks. Then we'll introduce you and then we can move ahead with your scientific talk. So this is how the procedure would be. And in the question answer sessions, we'll be picking up the most relevant questions from the audience and then we'll field it to you for answering.
Right.
So to begin with, again, here we go. Once again, I, Ajay Jha, on behalf of ACS India team, welcome you to this first ACS Science Talk session of 2022. Just to update you on the upcoming ACS Science Talk, please visit our ACS in India webpage and register for the upcoming sessions. We have also updated the past lecture recordings on the ACS Science Talk virtual library page. Feel free to visit, like, subscribe, and share among the scientific community. To keep you updated with the latest research findings, the products, and launches at ACS, we have a monthly newsletter, ACS Insights India. Subscribe today for free. We'll be able to provide the link into the chat box later. Again, it's an honor and privilege to have Professor Umapathy with us. And Professor Umapathy is a J.C. Bose Fellow and Director of Indian Institute of Science Education and Research, IISER Bhopal. He obtained his doctorate from the University of Otago and has also been SCRC Research Fellow of the Rutherford Appleton Laboratory in Oxfordshire and University of York. Professor Umapathy is a professor at the Indian Institute of Science since 1990. He leads the laser spectroscopy group at the Department of Inorganic and Physical Chemistry and Department of Instrumentation and Applied Physics at IISc. In addition, he also held a special professor position at the Department of Chemistry, University of Nottingham, UK. His research interests are focused towards understanding structure and dynamics of molecules using different spectroscopic techniques and deciphering structural uniqueness of molecular entities in complex biological systems. Professor Umapathy is the recipient of numerous awards and fellowship, to name a few: the prestigious Santya Swarup Bhatnagar Award 2004, Swarn Jayanti Young Scientist Award 2000, Holman Chair Professor from University of Groningen, and Raman Mishishuma Award from the India-Japan Science Promotion Council, among many others. He is also Fellow of the Royal Society of Chemistry, elected Fellow of the Indian Academy of Science, and Chairman of the International Steering Committee of Raman Spectroscopy. Professor Umapathy holds editorial positions in several journals of international repute. And before Professor Umapathy delivers the talk, couple of instructions for the audience. We request the audience to post their questions into the Q&A tab. Please avoid putting questions into the chat box, and in case of any internet issue, please stay with us. Now, I would like to invite Professor Umapathy to deliver the talk. I will stop sharing the screen. Yeah.
Yeah, sure. Yeah, you can share the screen. Okay, thank you very much, Ajay, and it's a pleasure to be here sharing some of our work in the last 10 years or so. So let me start saying that, lasers, the reason we call laser spectroscopy is that one can study fluorescence, emission, absorption, and reflectance in addition to scattering using light, right? So now if you look at lasers themselves, lasers have this unique property that other lights don't have, which is the coherence property, which gives this ability to have extremely pure frequencies that is controlled by the Heisenberg's uncertainty principle. The purity can be as pure as plus or minus 0.001 nanometer, which actually then leads to this property of being able to focus the light to the theoretical diffraction limit, which is half of the wavelength. So if it is 500 nanometer light, you can focus it to 250 nanometers, which is approximately lambda by 2. So the lasers, they provide a unique opportunity to explore science in a way that normal light cannot do. The frequency purity allows you to do extremely high resolution spectroscopy, which normally white light cannot do. And one of the high resolution spectroscopies is Raman spectroscopy that I will talk about in the next few slides. Now, when you actually put a light through a very highly pure light of specific wavelength like lambda 1. And this light interacts with the molecule. Let's say here you have CS3OH, which is methanol. And the distribution of scattered light that comes out will contain different kinds of frequencies. One frequency is lambda 1 itself, which is the incoming light. And that frequency is what is called Rayleigh scattering, when the frequency is the same. And then you also get light scattered with a different frequency. And this difference in frequency is related to the interaction of various electrons in various bonds, depending on how the electrons are polarizable in which bond. And the amount of energy required to polarize that energy is what is shifted in frequency lambda 2. Now, that shifted frequency then is a direct representation of the different kinds of bonds that you have in this molecule, like CC, carbon single bond, CH bond, or OH bond, or other frequencies which are related to different actions of the various vibrations. If you look at this is an example of types of vibration. If you take a 3-atom system, you see here you have the 2 atoms are moving in phase, which is called symmetrical stretching. Here, if you see this is called asymmetrical stretching. So in addition to that, they can also do bending, and you can have rocking motions. And of course, there is twisting motions, and you have wagging motions. Each of these motions have a frequency, and that frequency is interacting with the light of high frequency, can give you a scattered light representing these motions. So by looking at the frequency that we measure, we can tell you what are these atoms and what kind of motions are they experiencing. And then by looking at this in time, you can actually say why they are doing this motion at what time scale. So this is in general is the laser spectroscopy principle of trying to understand what happens in a chemical system when you do any processes, chemical or biological processes. Now, of course, we are talking about biology, so we must also look at the light-tissue interactions. And of course, these intensities, for example, of depth of penetration depends on the absorption coefficient and the scattering coefficient. And so we can calculate how far a light can go through in a tissue. For example, what you see here, in this picture on the right side, you will see that different wavelengths actually goes to different type of depth. Here you can go from 0.1 to 0.2 nanometer from the surface. And if you actually go to 1064 nanometers, you can penetrate up to 2 to 6 millimeter. So in biology and medicine, one of the biggest question is, the body has different levels of tissue depth. Can we actually go from the top end to the bottom? Therefore, can you look at different veins, different nerves, and different skin depths? And therefore, can you learn anything from that using spectroscopy, using this process of depth profiling, plus also studying structures using Raman spectroscopy? Of course, biology is very complex, and you can see a simple molecule, a protein structure has alpha helix and beta sheets. And inside those, you will have a chromophore. Let's say if you have a hemoglobin, you'll have a heme chromophore or a cytochrome. Now, if you actually take one of these helixes, you will see that there is, if you say beta sheet, you can see that there are different kinds of frequencies that corresponds to different bond movements, CO or CC, or you can actually see, this is called the amide 1 band. And if you actually go into the deformation of the CH and the NH in the amide group, you will call it the amide 3. So by monitoring these kind of sections, one can actually talk about the ratios of alpha helix to beta sheet. You can also calculate the quantum in terms of ratio of the volume of these two species, in addition to which one is most dynamic in a particular protein process of folding or unfolding and things like that. So this is what we try to do. Now you can ask the question when you can actually talk about only one particular frequency for a particular bond. If you take a cell, cells contain different components. If you take a skin cell, it contains proteins, it contains RNA, DNA, and lipids, and of course, membranes of different kinds. And of course, you can see if I record a Raman spectrum of a cell, this is what you get, frequency versus amplitude. If I record protein, you get a different spectrum. RNA gives you a different spectrum. So how do you study a cell? So luckily in the last 30 years, this has been the objective of many of the colleagues around the world, trying to understand in simple in vitro study of cells and proteins and RNA, or you can lyase a cell, separate the components, and then study them, so that we understand what these frequencies means in biology. So you see on your right, the peak positions are shown, 1660 or 1450, like that. And then you can look at what these represent, and therefore what is the component in biology. Is protein or lipid, and you can see nucleic acid or amino acid response. So you can learn broadly as to what are the chemical changes happening, in which part of the chemical component the chemical change is occurring. And that's what we are interested in studying. So now in my lab, we look at many, many different biological processes. For example, we look at tissue studies. If we look at the tissues here, we look at cancer tissues before radiotherapy, before chemotherapy, biopsy tissues, after chemotherapy and radiotherapy, biopsy tissues, in order to understand the level of cancer, number one, and also to understand whether the cancer is progressive or not. So that work is going on. And we also work on drug-cell interaction. And for example, if you're dealing with the cancer drug, and how do you know which drug is most efficient for a particular patient? And so we need to understand this at the lab level in vitro, and then we show in different drug lines how these things work. Similarly, we look at liver injuries, we look at sepsis and nerve injury where neurons are affected. You can neuromyopathy, cardiomyopathies can happen in muscles. We also look at bacteria, which we have patented quite a bit. We work on lab on a chip, plus also diffuse wave spectroscopy. But today I'm going to share with you some few examples of this, and that give you an idea of how these experiments are done and what do we learn from it. So one example I'm going to talk about is brain cancer cell and drug interaction. One of the drugs that is used in cancer cell is called the HDAC inhibitors, what you see here, which is histone deacetylase inhibitors. Why do we need that? If you look at our normal proteins in cells, what you see is these kind of histone proteins where the DNA coils around. And these histones are different types. One is a histone core, or it can be a linker protein called histone H1. So you can see here that it's a nuclear protein and DNA winds around it. So one can study can you hear me now?
Yes, we can hear you. Yeah.
So I somehow lost. Yeah, that's it. Yeah. Yeah. Go ahead. Yes. So you can see in the screen. So there has to be an equilibrium between acetylation and deacetylation process. because in order for the cancer cell to die, we need to make sure that balance is maintained. But most of the time, what happens in cancer cell, the acylation continues to happen, and that is why the cancer cell continues to grow. So the inhibitor allows it to de-acylation inhibitor, controls this acylation and de-acylation process, and therefore doctors have found many different kinds of inhibitors which are used for different kinds of patients. So what we do, we try to measure the acylation process. What you're seeing here on the screen, in the first one, you see what's called a valproic acid and sorbitol are the two different kinds of drugs, and one is a control. So what we do is we incubate the cancer cells for 48 hours or 24 hours and monitor them at different time interval. What you're seeing is a summary of this. And we also have sertinol is one kind, apicitine is another kind of medicine. And so we also monitor these changes in the CH stretching frequency and then decide what happens. What is strangely happened during this experiment was the biologists are aware of acylation and de-acylation, but in our experiment, we found propionylation. You see here, this actually shows you additional CH2 bands, which are related to the propionylation. Then we try to do this in various cell lines. You can see the different cell lines correspond to different kinds of human species, and we tried various kinds of drugs, and we demonstrated that we can actually understand efficacy of these drugs. Now, when I told my biology friend with whom I collaborate this project that we are seeing propionylation, initially he disagreed, saying it is not known in glial cells, and so therefore it is not possible. Then I said, let's repeat the experiment. We did, and then what he found was very interesting. He actually used an assay and identified propionylate histone in his control experiments. So then he was shocked to figure out that we can also learn something new from spectroscopy in the biological side. So now they are also looking at medicines which actually has to control propionylation in addition to acylation. So what have we learned from this process? We have learned that the propionylation can happen, and there is a correlation between propionylation and cell death. Larger the propionylation, larger the cell death. And we have also demonstrated that it's a fast, reliable way to identify the drug screening and also the cell line screening of a patient. So this is one example of how spectroscopy can help biology in learning something. So all these data have been published. So you're welcome to look at the literature. You'll be able to find these papers in the Journal of Biophotonics or Analytical Chemistry. Another example I'm going to talk about is muscle diseases. Muscle diseases, these are all neuronal induced muscle diseases. People will get affected. So all of you know about Stephen Hawking. And of course, Marathon King is a Miko True. So he's a cardiomyopathy. And Stephen King had a neuromyopathy, memory myopathy. Now, this somebody has to understand from identifying at the disease time, which is not so easy, identifying what disease it is. And therefore, there is a lot of questions about how to treat the patient when they come with a neuromuscular disorder. So my colleague was working on trying to understand this from a biological point of view. And therefore, he offered to discuss this and see whether we can apply Raman to identify this. And so what they do in medicine, when you're trying to study muscle disorders, they actually use Drosophila as a model system. The Drosophila muscles, what you see here, is equivalent to our human muscles, and it has a lifetime of something like 70 days. And so for a human muscle to evolve, it takes 10 to 20 years, sometimes 50 years, 70 years. So therefore, we can study using Drosophila as a model system, because the genome matches 75% is as good as human disease genomes. And we can easily study these fibrillar muscles by mutation, by inducing mutations in Drosophila, which you cannot normally do in a human muscle. So when we study the model system, what you're seeing here, ideally what happens in a human muscle, you see here, there are different, this is the way our muscles are connected. These are the angles in which you can stretch or you can compress. And these are the sections where nerves get extended. These are all controlled by different kinds of proteins. Actin is a protein, tropomyosin is a protein, and you also have troponin as a protein, myosin is another protein. All these can be mutated. So my colleague in IISc is an expert on this. So he now what he does is called, he removes the troponin, which is what we call EP1, and similarly he changes actin, which is removed, myosin is removed. So there are different mutations generated in the Drosophila. So these are removed, and then Drosophila was grown, and then we monitor their muscles every day from first day to 60 days. And then we learned the second day to the 12th day is where the most of the changes happen. So we recorded Raman spectra in those days. And what you see here, 2-day-old flies, you can see on the top, and the 12-day-old flies. And these are the spectra that we have recorded for different kinds of mutations. And these mutations tells you which part of the muscle is being affected and what are these mutations related to. And this can be statistically analyzed by using a cluster analysis, hierarchical cluster analysis. And what you see is the cluster analysis on the top and bottom. Now, by doing this, we can do some more analysis and tell you that, you can see that we can have a sensitivity of 89 to 88%, and specificity even go up to 98 to 99% in identifying different kinds of myopathies. So you can see that you can separate Nemelin myopathy versus cardiomyopathy. The Nemelin myopathy deals with UP1 and KM88 protein, and here what you see is cardiomyopathy is UP101 and HTP2. These two are different kinds of changes in the protein structure. Now we know by looking at the Raman spectra, you can identify which myopathy it is, whether it is Nemelin or cardiomyopathy. If it is Nemelin myopathy, then the UP11 or KM18, and I'll show you what they are. You see here, KM88 means that the actin is dysfunctional, and UP1 is troponin is dysfunctional. So that is Nemelin myopathy. So by looking at this, doctors can treat them by knowing which part of the muscle is now destroyed and therefore what they need to do. And so when I said this to my colleague and of course to the doctor was excited, and then he gave us a sample of a patient where we identified it as Nemelin myopathy, which he could only do it by empirical method. So now we are trying to now take samples from the hospital, trying to help the doctors to identify as soon as the biopsy is done. We try to help them to identify what myopathy it is, so doctors can start treating for a particular kind of protein deficiency rather than generally trying all proteins. So this is another advantage. This is also published. You are welcome to look at it, the publication. Now, coming to the last section of the talk, this is something very futuristic, so I wanted to share this with you, very recent work. We talked about depth profiling on humans of medical applications. So you can ask the question that how deep can we go? What can we learn from it? And so if you look at it, we have to start learning about how does photon propagate in a body or in a system. Now, so we try to understand this. How do we understand photon propagation through turbidge media? Is very well known, for example, in electrical engineering. And they use it to understand cloud movements, trying to measure frequency in the environment, and therefore they try to follow how photons can propagate in turbid media. Now, turbid media could also be, for example, tissues, which has very similar densities, but then there's a lot more scattering processes that happen. And as I said, you saw near-infrared absorption can go through various parts of the tissues. So people have done brain scans for neonatal babies to look at the oxygen concentration into the brain through the skull. And that is very well known. They look at the absorption of the near-infrared light, and then they decide whether there is sufficient oxygen flowing through the brain or not. And there's a lot of studies also about Rayleigh scattering studies, trying to understand what happens inside the body. You might have already come across our scanning in our bodies using light, mostly ultrasonic scanning. They are all frequency scanning. Of course, there are different kinds of frequency compared to light frequency. So you can do photon correlation studies, which is very well known. So having known all these, our question was, why can't we do this using Raman spectroscopy? Because we know that light penetrates at deep distances, but how do we identify it at a specific location? How do you measure the signal at various parts? So we started analyzing, trying to understand modeling. We did some Multicolor simulations and learned that light actually incident beam can go from one place and hit a particle. It can generate large number of Rayleigh scattering and some Raman scattering. The Rayleigh scattering again can hit another particle, which can generate Raman and Rayleigh. And the Rayleigh again can hit another particle. Therefore, the light can propagate in a multiple scattering mechanism. So therefore, we can use a multiple scattering process as a way to identify what happens. And then finally, you measure the signal at various, for this particle, you can measure them here. For this particle, you can measure the signal here. For this, so by going around the particle or the system, we should be able to measure. That was a theoretical, hypothetical idea. And we did this in Multicolor simulation. We could see it. Then we decided to get into the experiment. The simplest experiment we could do was this. So you take a glass vial attached to each other, 2 units. I put chemicals in the front unit, 1 chemical, another chemical in the second unit. And similarly, I take another vial with three vials, all attached to each other, 1 in the front, and there's 1 in the middle, 1 in the back. And then I try to put light only one side. Can we identify the chemicals which are in the back is the question. So we tried to do that. This is our first experiment. You can see here. The front part had trans-silvene. In the back part, we had 4-benzophenone. By imaging the outcome, you could actually see the front section of the vial was separately signaled and the back section signaled separately. It was a great improvement. We learned a lot. Then we decided, let's start looking at it in bottles. And so we went and did what can we see in different bottles in different colors. You can see here, this is a brown bottle without opening. You can actually put light through. And then you have this green bottle, which also can go through light. And of course, you have this white normal PET bottle, and they're all glass bottles. You can still go through inside and see what is containing inside the bottle, whether it is water or something else. And of course, hydrogen peroxide is one of the dangerous chemicals which can be used along with other components to create minor explosion. And so this is why you are not allowed to carry water bottles inside the aircrafts. And therefore, we demonstrated we can actually identify even less than 5% of hydrogen peroxide mixed in water in any bottle. And so this is good for, in general, for any explosive detection kind of applications. Having done that, the next question was, can we actually penetrate envelopes? And so we did that. You can see that airmail envelopes, green envelope, which people use some these days. And of course, this is your cardboard-based envelope. We can still see what is inside, whether it is a chemical and what kind of chemical it is. So now we are able to see, without opening anything, whether it is the front or the back of the envelope, we can still say what is a chemical hidden inside the component. Having seen that, this is ammonium nitrate hidden in the envelope. Having seen that, now next question is, can I see the diffusion of photon to many places? So then we put a vertical column of a tube. We pack this tube at the center part with ammonium nitrate, at the bottom with trans-silvene, at the top with dichlorobenzene. So when we did that, exciting research was, we put the beam a little bit here and then observe the light that's coming out and measure the signal. And this is what we started seeing. Depending on where the light is located, you could actually see what is happening. So if you put the light here, you see the photons migrate to this side, give the scattering here of the DCB. We can actually record the DCB spectrum along with the trans-silvene spectrum. This is ammonium nitrate and this is here. So you can actually measure at this point, you get this spectrum reasonably well. And if you put it at the center, what you get is ammonium nitrate. If you actually go down, then you can measure the surface as well, in addition to trans-silvene and DCB. So we are able to demonstrate that it is possible that you can see a mixture of DCB, trans-silvene, if you actually looked at it in vertically. But if you put it on the side this way, you can actually measure individually what they are. Having done that, we got more excited, more ambitious. And of course, we went into this idea of, for example, I hide a chemical at the center, one inside, and then the another set outside, and then measure the signal by moving the exciting light from top to bottom and measuring light from all directions, 360 degrees. And you can see here what we see as we move from one to the other. The center contains the material that we are interested in, which is ammonium nitrate. And therefore, you see the intensity start growing up and see ammonium nitrate maximum at the center and then start dropping. So now we can see in 2 dimensions what happens. The next question is, can we go 3 dimensions? Now that's what is called shape selection inside a hidden material. And that's what we did. What you see here is, for example, a chemical outside and another chemical inside. We try to then measure the distances and the width. We can then tell you exactly how far this chemical is hidden, what is the thickness of the chemical, and how far from the top, how far from the bottom. Therefore, we can restructure this. Then the next question is, okay, it is 1, it's okay. What happens if there are 2 units are separated in 2 different distances? You can see here, you can see the signals come out, and then you can actually measure and calculate how it is done. So then went on to do this. You can see more accurately with a 100 micron resolution. You can see the distances here is 0 to 50 millimeters, so 5 centimeters we can go. And we saw this at 100 micron resolution. We could clearly see the 2 lobes coming out, saying the chemical hiding inside the part. So then we decided to do this image processing using the CCD outcome. And the CCD outcome clearly tells you that by scanning at every point in 2 dimensions, in each of them is a hyperspectral plane. By scanning at every level, you have 100 micron and 150 centimeter. You can see 500 spectra. You can then reproduce what is inside and what is outside by doing mathematical image processing. And we can easily predict what kind of structure is present. Is it the same chemical or a different chemical? And that gave us the confidence to go further. Then what we did, we decided to go into a tissue which is about 12 centimeter long and 5 centimeter thick. And we drilled a hole through and put a chemical transstill beam inside in a chicken tissue. And then we did the experiment using scanning around the signal tissue. And we identified exactly the location of transstill beam and the size and shape of the pellet that we hid in the tissue. So now we know that we can penetrate the tissue and identify the chemicals as accurately as you like. So where does it leave us? It gives us the option to look at how far can we go? You know, can Raman spectroscopy, Raman imaging, can it become a tomographic unit? Can we convert the MRI into MRI is magnetic resonance imaging? Can this become a 3D Raman imaging? Therefore, in addition to learning the density changes in the liquids, can we actually start seeing chemical changes in the body? And therefore, you can see damages in tissues, damages in bones, anything else much more accurately, including cancer and the size of the cancer and all that. And that is our ambition, and that's where we are beginning to go. So all these technologies are all patented. What you see here is 4 US patents granted in the last 2 years. We are now going on and trying to do more important things. And this is, for example, is what the summary of what I just told you. This technology can lead to security applications, material identification in deep ocean, for example, forensic in people's in cases, courts and stuff, biomedical applications, of course, pharma industry for counterfeit, quality control, and process control. And I should say that these are all supported by my colleagues from various departments in biology and also colleagues in various hospitals. What you see here is the top 5 names that you see here. All of them are from microbiology, Somasundaram, Nandi from biochemistry, Annapurni from genetics, Sigdha from biophysics, Naktomba from genetics, Saini also is from genetics. And Santosh is a pathologist from a mental hospital in Nimhans in Bangalore. And Geeta Mukherjee is in Kidwai Cancer Hospital, and Kulkarni in the Ramya Hospital, who is a general physician. Of course, many of the funding, the industry funding comes from Boeing, ITC, Unilever, and Procter & Gamble. And of course, these are my students of different generations, have contributed to starting from basic spectroscopy to applied spectroscopy. And these are the most recent group of my students. The funding from various agencies like DST, DRDO, and DBT have supported most of our research in the last 20, 30 years. And I must thank you for your attention. And I thought this might go much longer, but anyway, I managed to finish in 35 minutes. So thank you very much.
So thank you, Professor Umapathy, for this wonderful and illustrative talk. And again, before moving to the question and answer session, we'll have a quick poll for the audience. So I'll launch the poll, and we request the audience to just post their response. So here is the poll. And the questions are: after attending the ACS Science Talks, how likely are you to suggest this to your colleagues? So recommend ACS Science Talks to a colleague, publish in an ACS journal, join or renew ACS membership. Are you attending ACS Science Talks for the first time? Yes or no? And then, which is the mode of communication? From where did you get this notification about the ACS Science Talks? So it could be around the ACS website search, WhatsApp, social media, emailers, from a friend or colleague, or any other medium. So we'll wait for a few seconds before we close this poll. So I'll begin the countdown. So 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. So we can see 64% of the audience have participated. I'll end this poll, and happy to share the results. So as we can see here, 67% of the audience would like to recommend this to their colleagues, 22% would like to publish in ACS journals, and 10% would like to renew their membership with ACS. Glad to note that 41% of the audience is joining for the first time in this platform. And again, emailers are the best mode of communication. Again, at the same time, the landing page, the ACS website. So thank you so much for your responses.